So welcome back, everyone. Here we are on the next to the last lecture, not only in the course of healthcare bioethics, but also in the program of bioethics, the Masters of Science in Bioethics at St. Thomas University, Miami Gardens, Florida. Beginning always with a little prayer in the name of the Father, the Son, and the Holy Spirit. Blessed Trinity, we love you, we adore you, and we thank you for your many blessings, Lord. In spite of all the difficulties of life, and particularly these past 2 years, by your grace, Lord, we are here today, alive and well, thanks to you. And we thank you for this. We thank you also for the opportunity of participating in this teaching and learning experience in the issues of bioethics that surround us. Precisely today, as we look at the bioethics of pandemics and the context of healthcare bioethics, we continue to pray for those people who are in most need of your divine mercy at this time. This we ask in the name of Christ, our Lord. Amen. Okay, as always, if there are any questions or comments before the actual lecture, feel free to speak up. Otherwise, we're going to move forward. And before I forget, so this is the last one, right? The last one will actually be on December 4th, Saturday, December 4th. And I think Chris, who lives up in Broward, is going to try to make an effort to be here live also. Jorge, please be here live also. And the carrot is that we'll have a little social after the lecture. All right, so around noontime, if anyone wants to bring anything to eat or drink, feel free to do so. No alcohol until after 5. But anything you want to bring, I'll bring stuff so that we can share something, okay? And just celebrate the end of the program as a whole, this cohort. And we're starting a new cohort in January, so let me speak about that for a moment also. We did not have a cohort in the fall, again, affected in part by COVID, because one of the main venues that I use for announcing the program is different conferences and workshops and conventions, and all that was shut down in 20 and even in the first half of 21. So I didn't have a chance to go around much and publicize the program. And actually people didn't want to come either, the classes being blended and all that. So for now, there is a search, and several candidates have already shown an interest and in the process of being admitted into the program. So long and short is we're going to start a new cohort in January, all right? So if you know of any family or friends or anyone who may benefit from this, let them know, please. Obviously, the best advertisers for a program or for anything, even a product or a service, is the people who have gone through the program. So satisfied customers, all right? If you're satisfied with the product, then please announce it and tell people to apply, because we'll start a new cohort in January. And the silver lining of that is that since we go for 4 semesters, then that new cohort will end in May of 2023. You see, in the spring of 2023, which coincides with the commencement. And so unlike yourselves, that now you have to wait 5 months for commencement in May of 2022, they will roll right into commencement the week after finals here, okay? So that's a silver lining there. We line it up with the fact that we only have 1 commencement ceremony now at St. Thomas University. When I started the program, we actually had 2. We had 1 in December and 1 in May. So people were graduating, the first cohorts were graduating right away, were actually having their commencement ceremony right away, but no longer. So new system, we're adapting. All right. And even if it's some persons who are not sure about the program or any details of it, you can give them all luxury of detail for sure, because we've gone through it. But also, if they have other questions, please give them my cell phone number, and I'd be happy to talk to anyone about the program. Okay, and that's about it. Okay, Now, pandemic bioethics. Talk about being opportune. In fact, this is a new lecture that was not here in the previous cohort because we were just going through the pandemic at that time. And so we did not actually have a lecture on this, but it's a new one that I have built since, especially getting data from the last year and a half on the pandemic, both on the clinical side and on the ethical side of things. We need to combine those for doing the bioethics, right? So first, a little clarification about the nomenclature itself. A lot of this is probably, oh, hold on, there's a little chat down here. What's going on? A minute, please. Oh, okay. All right. So you're up in Orlando. Wow. All right. Try to make it. Let us know, please. Thank you. All right. So a little bit about the nomenclature first. In other words, all the terminology that's being used. COVID-19 is actually the disease. All right. COVID, that acronym of letters, it refers to the actual disease, all right? Just like the flu, for example. The flu is caused by influenza virus. The virus itself, its technical name is SARS-CoV-2. And that itself is also a contraction, all right? That's the virus. The SARS acronym, SARS, stands for Severe Acute Respiratory Syndrome. So what happens with viral infections is that different viruses infect different organs. All right, and some are more systemic than others, meaning that some viruses infect several organs at the same time. Others are more specific. This one, thanks be to God, is more specific to lung tissue. However, lung tissue, specifically the alveoli within the lungs, are vital organs and vital tissues. So that's the downside of it, that it is a virus that can be lethal. Not all viruses are lethal either, okay? So there's a lot of just basic biological and virological clarification here to be done. Okay, so SARS stands for Severe Acute Respiratory Syndrome. And let me address the issue of syndrome because some people may not be as familiar with this word. A syndrome is a cluster of symptoms and signs and symptoms of the body, all right? For example, you've heard of Down syndrome, discovered by Dr. Down. That's why it's called Down syndrome, because that's the reference to the doctor who discovered this syndrome. Has physical and psychological aspects, a range to it, all right? It's a range of symptoms and manifestation. For example, on the physiological side, they tend to have a little bit of the slanted eyes, like Asian people. In fact, at one time it used to be called Mongolism because Mongolian people in their face look somewhat like that, rounded. They also have a particular characteristic that they curl their tongue, especially as babies, even before they can talk. They curl their tongue naturally. So it's another manifestation of Down syndrome at the physical level, all right? Sometimes they tend to be a little more short and squatty in size, but it's a range of physical characteristics from the most mild to the most severe. On the psychological also, you have a range of, from the most mild, that practically very little mental retardation to very severe mental retardation on the other extreme, okay? So there's always a range of this physiological and psychological characteristics, and any one individual with Down syndrome may exhibit any range of these, okay? So that's a syndrome. It's a cluster of symptoms and manifestations that are pointing to something, but it's a range. So that's what we're seeing with regards to COVID, the actual illness, the actual disease. What are we seeing? Well, we're seeing a range of things from very mild to very severe. For example, a temporary loss of taste and smell, right? There's a range there from very temporary, a couple of days, to months or years. There is also, on the more severe side, shortness of breath, again, from the very mild to severe shortness of breath, asphyxiating, where they actually need intubation, not just oxygen assistance, but actually intubation, all right, a vent, and so on. There have been a range of headaches, vomiting. Many symptoms are similar to a flu, because at the end of the day, it is a flu-like virus. It is an influenza-like virus, and in fact, it's a retrovirus with RNA instead of DNA. We'll get a little bit more into that. Okay. Also, the fact that it's called COVID-2, right? It's because, so COVID is coronavirus compacted, and 2 because it's a second strain that appeared in 2019, right? And there are a total of 6 strains of coronavirus. And the other 5 have been going around the human population for years, maybe decades. No one really knows how much, but evidently, they have not been anywhere near as severe as COVID-2, all right, from 2019. These other coronaviruses, these other 5 strains of influenza-like viruses have been around in the human population for decades, and they've been so mild that typically the typical symptoms are like a flu-like or like a cold, all right? Some runny nose, some sneezing, and so forth, but people keep going, and certainly it's not really recorded of a significant number of people who have died from those other previous 5 strains of coronavirus. Okay, so, but this one is particularly virulent. This is important with regards to its potential origin, because that's still debated whether it has a natural origin or a manufactured origin to this day. All right, it's controversial and certainly a big hot bioethical issue, especially if it was manufactured. Okay, all right, so this is the 6th strain of this coronavirus, and it did appear in 2019. Apparently, toward the end of 2019, it was reported in China toward the end of the calendar year, all right, around November, December. All right, now, I also want to point out that viruses have been around in the human population for millennia, for thousands, hundreds of thousands of years. And we have actually integrated them into our DNA, some of them, all right, are part of our genome. It's estimated that between 5 to 8% of the human genome are viral sequences or fragments of viral sequences. They're called HERVs, another acronym, human endogenous retroviruses. Retro, particularly, is a reference to RNA viruses. So you can think the R, retro, all right, is, these are RNA viruses instead of DNA. And that is also significant clinically and ethically, because retroviruses tend to mutate much more than DNA viruses, because RNA mutates much more than DNA. So let's take a look at that a little bit. This is significant that we have 5 to 8% of our genome, of the 3 billion base pairs that make up our human genome, are sequences of virus sequences. And that has to do with the cycle, how the virus regenerates or reproduces. Remember that a virus technically is non-living because it's missing the 2, so it's not a living organism because it's missing the 2 basic characteristics of life on Earth. Any living organism of whatever species, all right, has to have at least 2 functions, which is what? To be able to, if it's alive, if something is alive, something is metabolizing. It's metabolizing. You know, there's an organic process that is an orderly process, extremely complex, all regulated by enzymes, proteins, and other factors and so forth. but there's metabolism going on. There's metabolism going on in our cells, in our tissues, in our organs, and so we're alive. And that's the same argument for any organism that we see outside of these walls, all right? I don't care if it's a plant, an animal, fungus, bacteria, protists, you name it. If they're not metabolizing, they're not alive, they're dead, okay? Or they're not living. And the other aspect, it's necessary to metabolize, but not sufficient, because what happens? At some point, that individual organism is going to die, the individual organism, right? No matter how much we metabolize, we're going to die at some point. And so what happens is reproduction is the other characteristic to maintain the species, right? And so the other characteristic necessary for life is to be able to reproduce, to generate another one of itself, whether it's bisexual or asexual reproduction. And we covered that also way back at the beginning of the program, right? Okay, so metabolism and reproduction. Viruses by themselves are incapable of doing that, all right? They don't metabolize. In other words, they just have, either DNA or RNA encapsulated in a protein capsule of different design, which are very geometric, and they don't metabolize by themselves, okay? So they just kind of sit there. In that sense, structurally, they're closer to a crystal than they are to a living organism, when you think about it, because you have either DNA or RNA inside, which are very sophisticated organic molecule, macromolecule, and then a protein structure that can either be spherical or hexagonal and so forth, but they look actually more like crystals than they do like a living organism. And the other one is they cannot reproduce either, all right? And so they necessarily need to infect a host cell in order to reproduce. And in fact, that's what it does. When the virus latches onto a host cell, it injects its DNA or RNA and sequesters, takes over the replication machinery of the host cell, all right? It hijacks the replication machinery so that that machinery will replicate actually the viral DNA or RNA multiple times and make multiple copies. But in that cycle, there are two aspects to that cycle. Part of the cycle is that RNA or DNA of the parasite, it's an obligate parasite of the virus, in order to fool the replication machinery into replicating it, it integrates into the host DNA. It inserts itself into the host DNA, and that's also part of the code that is in that viral sequence, okay? And so when the replication machinery comes at its lightning speed through replicating that DNA of the host cell, it doesn't know, it just keeps going, okay? And that's what happens, that's how they reproduce. But then in that sequence, part of that viral sequence is coding for the proteins, for the endogenous proteins, for the proteins of the virus, the capsid, you see? And so it's a very clever machine, it's absolutely clever. That's how it does it. And then when many of these virus capsules and viruses manage to build themselves into that host cell, this whole cell swells and is full of these viral capsules, and then the cell lyses, it explodes. And when it explodes, it releases dozens or hundreds of these viruses from each cell. Each host cell will release hundreds, maybe thousands, I don't know, but I think it's the magnitude of hundreds. So each one of those viruses can then go to another cell of the host and do the same thing. So you get an exponential growth very, very fast. So typically within 24 hours of infection, the body is all infected with this virus, okay? So very, very rapid growth rate. And so you have those two phases, the lytic phases and the lysogenic phase. But part of that lytic phase is to integrate it into the viral DNA, the host DNA. And some of those viruses and sequences have stuck around, especially if they infect the germ layer of the host, right? Then it gets passed on to the next generation. So we've carried them around, and it's in our sequence for ever since we're a species. And it's not necessarily a bad thing either. They sit there, okay? They're called typically protoviruses. And what happens is that they actually get to stimulate the immune system to develop antibodies to that kind of sequence. Okay, so they actually are collaborating with the immune system to make us immune to these viruses that will have similar sequence out in the field, in life, particularly the retroviruses, because many of the retroviruses are these influenza viruses that keep mutating every season, right, year after year. So our immune system needs to expand on the basic sequence and fine tune so that it can kill also the new strain that just popped up this year. All right, so that's a little background. Now let's look at RNA and DNA for a moment, and why is RNA much less stable than DNA? Let's look at DNA first. We're starting with just the two-dimensional structure, the 2D structure, which is just a sequence of the base pairing, right? The rails and the rung of the ladder. Then as we put, and the base pairing, as we know, the ATCG base pairing. Then we take that two-dimensional structure and we make it three-dimensional, basically twisting the ladder, making the helix. So now we're three-dimensional. The first phase of the three-dimensional structure of DNA is the double helix, all right, the twisted ladder. But we know further that and that's the first level of coiling also. That coiling then goes around the nucleosome, goes around the histone proteins. Remember, there are 8 histone proteins, 4 pairs of proteins, of histone proteins that form like a little ball, all right? It's more closer like to a yo-yo. And the DNA winds around those that protein core 1.7 times, almost 2 times around, and then shoots out again and winds around another ball of histones. And this basic unit, then that is actually what is known as a functional unit of DNA, and it's called the nucleosome. Nucleosome because it's a little body inside the nucleus. Remember, soma is a reference for body, nucleosome. So it's the functional unit of DNA. And you have this going on a DNA string, which is billions of base pairs. Remember, when the DNA is in the nucleus, it's a single molecule. All those 23 pairs of chromosomes are all linked together into a single molecule that we call chromatin. All right. And so this is the functional unit. And this is like the second level of coiling, because the first level is the double helix. This is the second level of coiling, the winding upon itself. It turns out that these nucleosomes, which have been referred to as beads on a string, right? Beads on a string, like a pearl collar or something like that. They also wind upon themselves. They continue the coiling into what we call supercoiling, layers and layers of supercoiling. So it's more twisting and twisting, like taking a rubber band and twisting it more and more, making this clump. Compaction is the scientific name for it, compaction. So that supercoiling goes on into a fiber that is called the 30 nanometer fiber, because this has an average diameter of about 30 nanometers. Not to worry about that. But then that is kind of the basic fiber of chromatin. All right, and then that chromatin itself is also coiled inside the nucleus at the highest level, what we call the higher structure of DNA, of functional DNA itself in the nucleus. So it's coiling upon coiling upon coiling upon coiling, and that's why it's called supercoiling. And I don't know if you remember from back a year and a half ago, that whole thing is actually called compaction. And the compaction rate, like folding a leaf, remember how I folded a leaf? Maximum of 8 times. And by the time we get to 8 times, it's a little tiny thing like this. You can't really fold it by hand. You need a machine or something. All right. The compaction rate of DNA, of a 2-meter DNA single molecule, to get it into a super microscopic nucleus of a cell, the rate of compaction, everyone remembers? 10,000. 10,000 fold. All right. Compaction. You get 2 meters of a molecule inside a nucleus of a cell. Okay, so the bottom line about all this is that DNA is super stable, very, very stable. protected and so forth. So it's hard to get mutations of the DNA, but it does happen. You put enough UV there or enough heat or chemicals, mutagens, and you can get mutations in the DNA in spite of all this safety mechanism. In contrast to that, RNA is, first of all, is not a double helix, it's a single helix, right? So it's just the phosphate backbone, right? And then the single base is sticking out, just sticking out. And so this is a stretched out, already 3D structure, caricature of an RNA molecule, but it's not going to stay that way because you have bases that in that single molecule pair with each other. So the RNA is going to try to stabilize itself by base pairing upon itself. And so you have all these types of possibilities. Hairpinning, this is one of the most common one, all right? Or duplexes or loops and junctions and so forth. These are all computerized models of possibilities of a single strand of RNA with a random sequence of base pairs, of bases, sorry, how they're going to pair with each other. And it's a dynamic thing also. They go back and forth, all right, depending on the heat that's available, the salinity, and how some chemicals and some other factors make the molecule more or less stable, more or less dynamic to open up and close again. So it's super, super dynamic, totally contrast with DNA. RNA, at the end of the day, is very, very unstable. And that instability makes it a candidate for very easy mutations, relatively easy mutation, much higher mutation rate than DNA, okay? Maintaining the same sequence, but if you do, for example, a point deletion, if you delete a single nucleotide, what are you doing? You change the whole frame, you do a frame shift of the triplet, right? And it changes the whole sequence downstream for where the point mutation occurred and so forth. So they're much more susceptible to mutations, but because they normally occur in the millions or billions, if you have a mutant that survives, then that will generate typically first a new variant. And eventually when the variant gets, changes its structure somewhat, then it becomes a new strain, all right? So you understand in luxury of detail why RNA is much more mutagenic than DNA. Now, okay, this is back to the herbs. So we have two possibilities when there's an infection. It could be either somatic infection, meaning a somatic cell, which is the vast majority of the cells in our body, right, including lung cells of the alveoli, or it could be a germline. What's germline? Precisely the reproductive organs, testes or ovaries. If the virus infects the testes or ovaries, there's a possibility that it may incorporate into the egg or the sperm that is going to be transmitted. If that person reproduces, procreates, right, it's got the sequence of the virus integrated into its germ cell line or its germ cells, which are going to be the precursors of the ovaries, of the eggs or sperm. So it may be passed on to the next generation. If that happens, there's a possibility that all of the siblings of the next generation will carry that sequence in its genome, okay? And that's how it happens. So here's a diagram just giving much more detail about the various protein sequences, or these are probably just polypeptides that form the actual envelope of the virus. Okay. Now, that's also because now we have, what is the delta? It's a variant, it's not a strain, okay? And now we talk about the mu variant and another one that hasn't even been named. So we're getting these variants that are cropping up. In fact, it's estimated that the original SARS-CoV-2 that left Wuhan, China, and went to Europe, by the time it got to Europe, all right, it had mutated already once. So that was another variant already. By the time that mutant, then that mutant went two ways. It went horizontally to the US, all right? And it went vertically to India and Africa. And so by the time it got to the US, it had mutated again. So probably we got the third strain of the coronavirus, that of the original one that appeared in Wuhan at the end of 2019. And then from here, it migrated south to South America and mutated again. In India, it went south from Europe, it went south to India, and then back up to the UK as the Delta variant. And then from the UK, it came over here as the Delta variant. So, you see? So this is what happens. When the RNA mutates, that becomes a variant. But when that mutation is significant enough to change the structure, then it becomes a strain. So there's more change, all right? And it could be enhanced. For example, it can have more sites of attachment, as is here. This is showing the spike protein, which is the site of attachment for the in the alveoli, all right? It can actually produce a variant spike that is more virulent, and that's why the Delta strain is more virulent, because the spikes apparently attach better, more efficient, and so forth. It's not necessarily more, it's more infective, but it's not necessarily more virulent, okay? But it's successful in attaching, more successful than the previous strain. All right, so that's the issue between variant and strain. Again, just some basic virology here. Again, if you have questions or comments, just jump in, okay? Now, let's look at herd immunity. Herd immunity is very significant and very important, and it's achieved experimentally, all right? But we have to understand the theory behind herd immunity, because we are mammals, biologically speaking, and the typical scenario for mammals is to cluster in herds, precisely. We call it socializing. So this is what happens with no herd immunity or herd immunity. The large individual here is the infected focus, the focus of infection. When this individual is interacting with people around him or her, is infecting the others around him or her, all right? And by the way, the infection, the incubation period for this coronavirus is 5 to 7 days, so about a week. What does that mean? It means that a person may be infected, but has not shown symptoms yet, or very mild symptoms. I may think just having a headache that night or that day, but it's actually a focus of infection for other people around, all right? So that's the incubation period. Once the symptoms manifest, it's been 5 to 7 days already. So now you have to start doing contact tracing to see in the past 7 days who this person was in contact with, which becomes a real mess, especially since we didn't know the incubation period or just anything about this virus that showed up all of a sudden at the end of 2019. All right, so without herd immunity, each one of the infected individuals will infect others around them at a rate of 3 to 5 persons. That's the infection rate, okay, that needs to be determined experimentally. Each virus will have an infection rate that is characteristic of itself, okay? 3 to 5 individuals, it depends on many factors, but each one of these individuals then becomes another focus of infection for the others around them. So you can see again that geometric expansion going on. With herd immunity, what happens? The green individuals are already immune. There are 2 ways of becoming immune, right? Natural or artificial. So the natural way of becoming immune is getting sick and surviving, having survived the infection, which, by the way, what is the survival rate for this creature, for COVID? It's around 97%. In other words, about 3 to 4% of people have died of COVID at the height of the pandemic, all right? And that's a very robust number, because it's actually millions of people when we look at a pandemic. When we look at worldwide data, all right, global data, it's a very robust number. So let's say about between 3 and 4%, which means that 96 to 97% of people infected with coronavirus will survive on average. It's a statistical number, all right? The problem is that we don't know individually, because we're not a statistic, we're a determined individual. So we don't know until we're infected and actually survive the infection, we don't know which side of that 90% we're going to be individually. We can be on the 97% or we could be on the 3%, all right? So until the event actually happens, we don't know individually, but we can give a statistic. So we can say that it's a good assurance, there's a good likelihood on average that an infected person will survive. Then we can start parsing the data and look at the general population and look at subgroups within the population. Obviously, some are more at risk or more vulnerable than others. And that's when we get into the elderly or the immunocompromised, et cetera, okay? But it's important to understand the principle of herd immunity. Obviously, the other way of reaching herd immunity is through the vaccine. Because even though the method is different, right, the end result is essentially the same. If I survive the infection, if I survive the coronavirus infection, then I develop immunity. I survive because I develop immunity to it. If I get the vaccine and survive the vaccine, I also develop immunity, okay? And by the way, the vaccines, at least the first 3 that came out, which we'll look at in a little while, give an average of about, let's say 90 to 95% effectiveness of immunity, which is really quite high for an influenza type vaccine. So that's herd immunity, we understand it. Now, the thing is, for each virus, the herd immunity number, the percentage is different because it depends on the infection rate, all right, the rate of infection. And so it has to be figured out experimentally. You've heard the 70% number, but how does that 70% come about? Meaning that if 7 out of 10 people are immune, then we've reached herd immunity. The likelihood is that the immunity will die. All right, that the immunity will die. Sorry, that the infection will die, will not be transmitted because 7 out of 10 people are already immune. So on average, if I'm in contact with that, with a person who is immune, they're not going to get it. Or if I'm in contact, or if they get it, they're going to survive, they're going to have mild symptoms. And if I'm in touch with or in contact with 3 out of those 10, right, that were not immune, then that person on average will have, on average, 7 people out of 10 people who are immune. All right, so if it doesn't die on the first round, it will die on the second round and so forth. So it's unlikely that the virus will then be transmitted with herd immunity. Okay? But there's that magic number. There's a magic number of 70%. So how is that reached? I said that that's reached experimentally because the infection rate varies from virus to virus. And that can only be tested. We don't test for it, we actually record the data. And we see on average how many people are getting infected with this particular strain of the coronavirus, all right? And it was determined already early on in Wuhan that the infection rate ranged from 1.4 to 5.7. So let's take the whole number average there. From 1 to 6 people. From 1 to 6 people were getting infected on average. For every infected focus, that focus of infection, that individual tended to infect a range of 1 to 6 other persons. So what's the mean there? About 3, right? Between 1 and 6, 2.5 or 3, or so actually here they have it at 3.5. So this curve is just a logistic curve of the threshold, which would be 1, you know, statistically 1 is 100%, right? So the scale is from 0 to 1 or from 0 to 100%. We draw a vertical line on that average of the infection, which is also known as RO or R null, that number. Go up to the curve, and then horizontally will give you the percentage. So here it's just under 75%, so it's about 70%. That's how we come up with this number experimentally. This is a standardized curve. And what is important, this is a number that needs to be found out experimentally for each viral strain. What is the infectivity rate? Okay. All right, so that's herd immunity. It was established early on. And now let's do a little bit of number crunching with that to see, without vaccine, how many people in the population and the absolute number of individuals need to be infected and survived in order to reach herd immunity. Okay, so let's do it globally. And this is August, so we have to go back about a year, because at the height of the pandemic, which was basically from May to in the middle of 2020, right, was the height globally. The thing was spreading, raging throughout countries, about 200 countries in the world. I was actually tracking this with undergrads during the Summer Research Institute here on campus, because we had to do it virtually. And this was data that we could gather data virtually, globally. The data was being crunched out daily from different countries of the world, reporting the number of dead, the number of infected, and so forth. So those are two numbers that we were getting from the global community, from all the countries of the world. The number of infected, number of dead. All right, so in August of 2020, they were these number of dead divided by the number of infected, okay, gave us the percentage that died, and that's the 3.7. So that's how we get between 3 and 4% death rate of this virus. Okay, the 100 is just to make it a percentage. So this is the number of dead on August 7 of 2020 worldwide, and this is the number of infected. You can see that the numbers are still relatively small compared to what we have today, because this data is over a year old, but it was kind of the height of the pandemic then. So between 3 and 4% death rate. All right, at that time, the human population was just under 8 billion, okay? So herd immunity is 70%, right? Or 0.7. So if we multiply the human population times 0.7 to see how many people need to be infected and have survived the infection to reach herd immunity for the world at the global level, all right? That's the number of people that need to be infected and have survived. So 5.5 billion people need to have been infected and survive to reach herd immunity globally. Follow? All right, now, how many of these would die statistically given the 3.7 infection rate? Well, we just take that number and multiply it times 0.037, which is the decimal of this rate, right, of this percentage. And that's the number of people who would have to die globally in order to reach herd immunity. 200 million people would have had to die to reach herd immunity at the global level, all right? It's a theoretical exercise, but 200 million people, that's a lot of people to sacrifice to reach herd immunity at the global level. This is without vaccine. That's why you see the urgency and the rush to try to lower this number, because that's what nature is going to do, okay? And remember, nature never forgives, okay? Not because a country was a very holy country, like the Holy Land, was going to be forgiven. No, Israel got it just the same as everybody else got it. All right, so yes, now backtracking, let's see what 200 million people represent of the world population, right? So we take 200 million divided by the world population number and multiply by 100 to get a percentage. That represents 2.6, almost 3% of the world population, okay? About 2.5% of the world population would have had to die to reach herd immunity. That's the bottom line of this argument. So that's a lot of people to have died for reaching herd immunity. The way to bring that number, of course, again, is with the vaccine and other means to try to prevent the infection to begin with, all right? To lower those rate of infection with the lockdowns and all that. Now, let's do a little comparison because we're calling this a pandemic, but there have been other pandemics in the world in the past, in history. The two most significant ones have been in our time, past decades, has been the HIV pandemic and the flu pandemic 100 years ago that you keep hearing about, the Spanish flu and so forth. All right, so let's look at those numbers. At the height of the AIDS pandemic, which was in the early 2000s, okay, the death toll globally was about 36 million people. 36 million people in the world at the height of the pandemic of AIDS were dying of AIDS, mostly in Africa. And at that time, the human population, HP human population was about 7 billion, okay? So when we do that percentage, we come up to 0.5% of the world population at that time died from AIDS. That's pretty significant, okay? Nowhere near 2.6, that's 1/5. So 2.6 is 5 times greater, the rate. How about the Spanish flu? In quotations, because it actually started in Switzerland, or is contested to this day where it started, but Spain was very hard hit because it's between the two world wars, right? And Spain was under dictatorship domination, it was locked down, and the whole country was very isolated from the rest of Europe and the rest of the world. So it was just a big incubation. It was a national incubation scenario. The death toll was between 20 to 50 million. It's still hard to say today how much, but even if we take the upper limit, it would give between 1 and almost 3% of death. At that time, the human population was approximately 2 billion people. So when we do the math there, we come up anywhere, a comparable rate, comparable with the COVID death rate for herd immunity today. Of course, back then there was no vaccine for the Spanish flu, okay? So they just had to take it in the stocking and absorb the shock, which was about 1 to 3% of the world population actually died from the Spanish flu. And that is an influenza virus, which is coronavirus, is an influenza type virus. So basically, this corona type virus is doing in nature what it's supposed to do, behave like an influenza virus, like a retrovirus, right? The vaccines for the flu virus that have been around for years and decades, and that we're supposed to take that booster shot every fall, right, at the beginning of the flu season, which is the winter, these vaccines are a range of 40 to 60% effective, because these vaccines, again, done in the traditional way. Up until 2019, we did not have the novel technique of messenger RNA, of mRNA, okay? So up until then, it was the traditional way, which was what? Use either the fragmented sequence or an attenuated sequence, right? The kill virus or attenuated virus to infect host cells and to develop the immune system that way and to tweak it so as to get the vaccine that will stimulate the immune system to do the same. And so that procedure takes normally 2, 3, 5, 10 years to do. So the flu vaccine, the flu shot that I'm going to get now in the fall was developed on the strain that was around 2, 3 years ago, which is not the strain that is around today. But hopefully today's strain will have some of the sequence of the strain of 2 or 3 years ago because it's a derivative of that one. So the vaccine that I'm taking today was designed for that virus, for that strain, hopefully will have some effect in today's strain. And that's why the average is only about 50% effective, okay? Because it's based on a strain that already is passé, went through already, 2 to 3 years ago on average. So that's why this novel technique of the messenger RNA that can be done within a year, within months actually, is so effective, 95%, because it's based on the current strain. Well, just maybe a previous version of the current strain, right? All right, moving forward. So up until now, we've seen mostly just biological, virological, and clinical stuff about the virus. But definitely there was a sense of urgency, global urgency to develop this vaccine, and millions, billions of dollars actually was poured into the research to get the thing done, to try to lower the death rate so there wouldn't be 200 million people dying from the just unleashed virus on the world population, okay? So many bioethical issues arise from there, okay? A whole panoply, a host of bioethical issues arise, and I'm just listing some here because they're still open questions. You know, how is it going to be produced? Precisely this novel, was the method compromised because of the novel aspect of the technology and trying to get it done quickly in months instead of years? Were some safety factors compromised, right? The trials, were the trials done sufficiently statistically significant on a population for the results to be robust, to be significant statistic? I can tell you that the population was minimal, did not meet the upper standard of getting certified by the FDA. What's FDA? Food and Drug Administration. They're the ones that certify, give the clearance for any food that we eat and for any drug that we take, any medication, all right? Whether it's injected, orally, you name it, it has to have FDA approval. But there are two levels. There's an emergency approval, if you will, because it has not met all the conditions for a permanent approval. And that's what happened with these vaccines. They were giving emergency use precisely because of the critical need in the population, the US population and the world population. So they were given those three, the first three vaccines that appeared, right? Pfizer, Moderna, and Johnson & Johnson were just giving emergency approval, but not final approval because there was, the data was not very robust, really. They had done only a few trials, not significant, not statistically significant to secure, to give good assurance to the population at large that it would not have side effects. And sure enough, side effects started coming up, right? That became part of the data eventually. What does that mean? It means also existentially that all of us who were getting that flu vaccine, including myself early on in 2020, 2021, was really an experimental vaccine, okay? So we were guinea pigs, we were experimental subjects. So did we sign forms? Did we sign consent? Yes, I signed about 20 forms to get that vaccine. And was I reading it? No, because I just wanted to get the vaccine. And I was taking, I already had preconditioned myself to take the chance, to take the risk, okay? So whatever they were asking me to consent, I consented, right? Was it informed? Well, it was on me. They gave me the information, but it was all part of the 20 pages of signed documents. Whether I took time to read it or not, it's up to me. It's up to the individual. But we were essentially all guinea pigs, part of the trial, to get the data to be as robust as it is today. Okay, so there are ethical issues there. Then at the beginning also, who gets it? Well, the vulnerable populations classically are the elderly because our immune system gets worn out, all right? And also the immunocompromised, that's a no-giver, a no-brainer. For example, people who have transplant patients, some friends of mine have had transplants, and they are on immunosuppressors. So obviously they're compromised. What is a common cold for us, for them can turn into pneumonia because they have fewer, their immune system is not shut down totally, but is attenuated, all right? But then the third group that was a surprise classically is children, because their immune system is not as developed as an adult. Of course, they haven't been exposed just simply the number of years that they have been around. It's not enough to have been exposed to a variety of stuff, viruses and bacteria and so forth, to develop a robust immune system, right? They're still growing. However, surprisingly, at first, the children were not getting infected. Were actually getting infected at a lower rate. And if they were getting infected, they were showing milder symptoms than adults. And that was a big conundrum at first with the scientific community. And eventually someone explained it to me, actually a pulmonologist said, perhaps what is happening, what do you think is happening? It has to do with the specificity of the virus and the infection site. And because this is a SARS respiratory virus, right? As opposed to digestive or circulatory or the heart or the brain or any other, the liver, you know, any other organ or systemic, the lung, the lining of the lung, the alveoli. It turns out that children's lungs, because they're actually physically smaller, have fewer sites of attachment. So actually have fewer sites, quantitatively, fewer sites of attachment than an adult lung. So the viral load, what we call the load, right, would be more effective on an adult lung because there are more sites of attachment. So now it's making sense, you see, but there's gotta be an explanation. There's no magic here, right? There's gotta be a biological, physiological explanation, just like there's gotta be a biological explanation when the human species started as a whole. All right, so all this is in the background of the bioethical questions, right? Who pays? Well, it turns out that it's free to us, but it costed billions of dollars to get this vaccine out. So countries had to take the hit. They had to take the hit in their budgets. Okay, so this increased the national debt by quite a bit. But then now the return is that this can also be sold to other countries and so forth, or bartered with the reduce our international debt and things like that, and it gets to the economy. So it gets really, really complicated when we go up into the international economy and trade and so forth. Invariably, who gets the short end of the stick? Developing countries, underserved populations, minorities, et cetera, we know it. And so this is the same with any other system that lives on the market economy. The ones who are at the margins, who are living more day-to-day existence, they're the ones who are going to suffer the most, have less accessibility and so forth. I can tell you here in Miami-Dade County, because I live in Miami Gardens, which is a low-income zone in general, there were big efforts made from Dade County Public Health Department to put sites specifically in low-income areas, you know, because the high-income people had cars that worked and everything, and they could get to the sites. But in here, the buses were shut down because there was lockdown and social distance and all that, you know, prevented many people in the area who are accustomed to walking or using public means of transportation. So there was a special effort concentrated on here in this area as one example, okay? So all those things have bioethical implications from the most local to the most global of the issues, ethnicities and so forth, where they actually divide or unite groups. Well, again, if it's going to be more on the market economy type of thing, then there's going to be winner or losers. Okay, and we have to then go into, it cannot be just strictly a market capitalistic economy. It has to be what we call a mixed economy. In other words, regulation. The regulation by government to make sure that the underserved populations have a certain priorities so that they level the playing field with regards to distribution, access, and so forth, okay? So many issues. There are still open questions today on, with regards to the bioethical things that can be raised with regards to vaccines. We're gonna pick up vaccine again toward the end of the lecture, right? But now we're gonna take a different tack, which is look at the origin of the virus, and some evidence, at least, as the thing was developing, because this is data that I've been gathering for about 1.5 years now, precisely since it started back in December, or at least there was public aware of it, public awareness of it. And the basic question here from the bioethical perspective is, was the virus natural or manufactured, okay? that you can see has huge bioethical implications, because if it was manufactured, then the next step is, are we talking about biological warfare? Which is something novel, that even we have gone as far as chemical warfare with Saddam Hussein and the Turkish population over there, his country and so forth, but not biological warfare, okay? But there is, we know that there's potential for it. There's evidence of stockpiling and so forth. And I can tell you, a nuclear warfare looks like a child's play, like kindergarten, compared to biological warfare, all right? Okay, so maybe this could be a good time to take a little break before we take a deep plunge into the question, was the virus manufactured or not? And I'll present my evidence, my opinion. And then you can do whatever you want with it. So let's come back just under, it's about 10:40 right now. So I'm gonna start in about 15 minutes, okay? Just under 11, and then we'll plow through the second half. So I'm gonna, questions, Jorge or Chris, anything? Comments, questions?
I do, but I think I'll save it for the end.
Okay, thanks. Let's see this one little chat here. Okay, thank you. So let me pause. If I remember how to do this, it's in here somewhere. Pause. Okay, welcome back. So here we go into the second half. Any questions or comments? Chris, you want to try your microphone, or see the tile here for a moment? So you had sent me a text earlier that you were asking if we could hear you. I don't know if you're back on your computer yet or not. Well, you can send me a text if you want, or a little chat, okay? I'm gonna get started. So the basic question is, was this a natural infection? There's a chat, let's see. Mayor, please. Okay, well, sorry, as long as you can hear me and the recording is going on, but if you have a question, well, I'll open my cell phone. If you if you have a question or a comment, you can always call me on my cell phone. I have it here, okay? Okay. Right. It's undisputed that the coronavirus 19 started in China, in the Hubei region, and in the city of Wuhan, which just from these maps, you can see the number of infections. Yes, yes, it's recording. It's a little dot there. Yes, thank you. Right, so just from the numbers, we can tell that the number of infected, right, and the number of dead, that it started in this region called Hubei and the city of Wuhan, actually. Now it's been identified, and the claim is that it started in this wet market. And before we go into the whole concept of wet markets in Asia in general, other places of the world too, we can see how China locked down internally, all right, but not externally, already in January of 2020. So we have to project back almost 2 years now I don't know where you were at that time, but think about January of last year. We were basically on lockdown. I mean, the thing was just coming up, right? We can see that the number of cases of infected was diminishing in China. Then it had a spike. Here is, this is actually, these ticks are on a daily basis, all right? For example, here is February 13. So this is European data, and that's why it's first a day and then the month and the year. So you see 2020, February, which is 02, and the date. So on 13 of February, China peaked, and then it declined very fast to a base rate just above 0. Whereas in the rest of the world, the infection rate was doing the opposite. It was doing a exponential growth here and just the opposite. But the focus of infection was in China. So you would expect, in principle, these two curves to parallel each other and to have the same kind of slope. Instead of a negative slope, to have a positive slope, certainly throughout China, because China has about 1/5 of the world's population, okay? And geographically, any part of China, geographically, is closer to Wuhan than any other country of the world, okay? So just by proximity. All right, so this is a site that is very interesting because it shows actual aerial photographs, satellite pictures of how China locked down from, hold on, give me a minute. I'm trying to grab it, highlight it. There. Messed up today. Copy, there. Website. Yes, from a mixture of nitrous oxide, which is a product of combustion, of transportation. This is the graph I want to show. Actually, satellite, this is satellite imaging of China. And this is what it looked like. This is nitrous oxide emissions. You see here the scale down here. This is December 31st, and this is January, the month of January. Okay, so the first half of January. This is the nitrous oxide emission of China measured by satellite imaging. Nitrous oxide is a product of combustion of the gasoline motor, all right? So it's what's coming out the exhaust. So this basically tells us that there was a lot of transportation going on in China in this time. And so people were moving around, basically, a lot. Then when we span this over, you notice the bar down here is going to move to February. So just 1 month later. That's what the nitrous oxide emission looks like at the end of January, beginning of February. Dramatic decrease. What does this mean? This means basically that China shut down, that people were forbidden from going out. It's a national lockdown in January, okay? National lockdown. Wuhan is here, pretty much in the south central part of China, okay? So again, December, January. Okay. Satellite imaging. So that represents this. However, internationally, China did not shut down. And these are news clippings from the time, all right? Here, for example, this is February of 2020. The Guardian. The Guardian is a UK journal or newspaper. It's a newspaper in Great Britain, okay? So, the Chinese government was upset that there were restrictions put on travel of people in China traveling abroad, because the international community was still trying to grapple with this new infection of a coronavirus, which was not really known exactly what it was. But what was known is that people were getting seriously infected with this and dying from acute respiratory syndrome, okay? So, I remember, in fact, at that time was a previous administration. Obviously, Donald Trump was the president of the United States at that time. And the president of China was complaining to the international community that the other countries of the world were discriminating against China because they're not allowing Chinese people or any person from China to go into other countries, international travel. And still, China did not shut down its international airport. Basically, it allowed people to leave China, even though nationally they locked down, because they knew they had a problem on their hands with this epidemic, and yet they allowed it to spread. They allowed flights to go out, international travel. In fact, 3 months later, or 4 months later, excuse me, even after the ban that President Trump put on flights from China, still about 500,000 people came from China in 4 months. Why? Because they were fleeing the epidemic. They were fleeing the infection there, and they came to the US, mostly through Pacific, because the Pacific coast of the US, we know, has a high population of Asian people, all right? And so in 4 months, 500,000 people came from China into the US alone, okay? Not to mention Europe. In fact, in Europe, what happened was that right around December, there was a fashion show, a fashion exposition of high fashion in Milan. I don't know if you remember back then, but Italy was one of the first hotspots in Europe. Okay? And Milan, particularly the city of Milan, which is in northern Europe, northern Italy, it's a big industrial city. And there was some kind of fashion design, international exhibition and show. And China being one of the major producers of textiles today, including silk and expensive clothing, right, was participating in that fashion show. And there were a significant number of Chinese delegates who had come to that show to expose their textures and to make textiles and to make contracts and all that. It was a high-end thing. And immediately, Milan lit up like a volcano of infections. And then that infected the rest of the peninsula. Eventually it went to the UK and the rest of Europe. So Europe was the second one to get infected. And then soon enough, it came from Europe over here to the US. So first there was that horizontal transmission going west from Asia into Europe, into North America, US and Canada. So now we have to do a little bit of tracking here, and the possible was known as zoonotic transmission. Zoonotic transmission is when a virus from an animal jumps into the human population. Okay? And we had that, for example, precisely with HIV. Before it was HIV, it was SIV for simian. Simian immunodeficient virus, okay? And then it went to human immunodeficient virus because it started in monkeys. This virus, the reservoir of HIV was in monkeys in Africa and Congo. And in the 1920s, somehow it jumped to a human from the monkey, all right? And that's called zoonotic transfer. It happens periodically from animals, the human population gets infected. So it's one of the first articles that came out, July of 2020, tracing the coronavirus 2 to a bat reservoir. Okay, particularly they're called horseshoe bats because the way the face looks kind of rounded. And these horseshoe bats are quite abundant in China. And they're quite abundant in their markets. This is wildlife that is being sold in China for decades now. So let's consider this for a moment. China has about 1/5 of the world's population. And of course, the demographics is a huge issue, right, for the government, how to feed these people. And so I understand, some Chinese people actually told me this, Chinese people who live here in the States, that during the Mao Revolution, okay, in the 1950s, there was not enough food to go around to feed the whole population. And so actually the communist regime encouraged the Chinese population to eat wildlife and to domesticate wildlife and so forth, and to go out into the countryside and just eat wildlife. And it's already part of the tradition because there were many rural areas that would eat wildlife, all right? It was sold in the markets like this for decades, and these are the wet markets. As you can see, this fellow here is wearing boots for a reason, because it's wet down there, okay? And these wet markets, the food is open like this. Carcasses, a lot of meats being sold. Here you can see the bats. They're fried or something, or cooked and sold. This guy is choosing one there. It's not cold in these wet markets, okay? It's a tropical environment. This fellow is wearing shorts and t-shirt. So it's warm in here. This food is there days on end. This is the actual Wuhan market that was the source of the infection. Supposedly is ground zero. And the name up here, very clearly, it says Wuhan, Huanan, Xiangxiang, Pifa. And so I Googled this, all right? In fact, before I go there, this is actually during the height of the pandemic. This is one of their ambulances coming out, and they have already cordoned off the area, as you can see, with barricades and so forth. And they're hauling out sick people from the market. So I just Googled this, right? Where are we? Here. So here's the Google search. And sure enough, it's a wholesale seafood market, and they have all kinds of foods in there, raw, live, you can buy live animals also. There's a little article that appeared in Time magazine, a comment. Ultimately though, experts say that it's hard to downplay the problematic nature of wet markets. This is throughout Asia and other parts of developing countries. They're named that way because of the large quantities of water that is used to slush the floors and to try to clean the place at the end of the day, all right? And so that soup of water that circulates through the floor is a mixture of organic products, right? Bodily fluids of different kinds mixed in with the blood and of animals and so forth, and other secretions of the animals. And so that's really are ideal opportunities. They just become cultures for viruses and bacteria to grow there and to thrive. And again, it's typically a tropical environment, so it's conducive toward these outbreaks of infections. All right, this has been known for decades, really. Now, it's not that we don't eat, buy, and sell raw food in the US, for example, okay? I don't know if you like sushi. When I was told about sushi the first time, I said yuck, because it's raw fish, right? So who wants raw fish? But then when I started tasting sushi, I love it. And now I love sushi. But of course, this industry is highly regulated. Highly regulated, and these sushi bars are inspected all the time. First of all, the food is almost frozen. You see these bars of frozen liquid that they have in the sushi bar. It's all glass, transparent, so you can see through, and whatever is left over at the day is thrown out at the end of the day. It's not preserved from one day to another. I mean, the whole industry is highly regulated, okay? And it's safe, generally. To eat raw food here. For example, we eat wildlife in Florida. We have gator steaks. Gator, I don't know if you tasted it. They say it all tastes like chicken, right? But gator is wildlife, obviously, and it's sold at there are gator farms for the meat and for the skin, the hide. Bison, I don't know if you've had bison, otherwise known as buffalo. It's very lean meat. It's about 99% lean, fat-free, you know, so it's very lean meat. Wildlife in general is lightweight. Also venison, which is just a fancy name for deer. So all that is sold, it's aged and everything, but it's very regulated, highly regulated, so that it's safe to eat the stuff. Okay? So it's not that we don't eat wildlife, but it's very different from this kind of scenario. Very different, okay? And it's all regulated with regulatory agencies run by the government, who themselves have their own checks and balances. So it's not that we would say an absolute prohibition of eating wildlife, but it has to be done in a decent, healthy, clean way. Otherwise, we're just exposing the whole population to outbreaks, these outbreaks of epidemics. Another possible source, this goes back a year ago, another possible source that came to mind was also traditional Chinese medicine, which also uses wildlife in a different form, snakes and lizards and other reptiles and mammals, as you can see here, but also herbs and plants. Plants also are subject to viral infections, okay? So another potential source back then, a year and a half ago, could have been traditional Chinese medicine doing the zoonotic jump from wildlife into human population, okay? But this one didn't really get much traction. Eventually, you didn't see much about this in the news. So I can say we can pretty much safely discard that it jumped from traditional Chinese medicine, but it's just another source of infections, right? Zoonotic infections jumping from animals or plants into human population. Another point I want to make, that China is not really, you know, a developing country, or as we used to call them back before political correctness, a third world country. Not at all. This is a highly industrialized nation. Again, it has 1/5 of the world's population in it. Wuhan itself, these are photographs of Wuhan. It's not a little rural backyard, little town, you know, with shacks. It's a megalopoly. It's got 11 million people in it. It's got this high rises, all right? This is the city of Wuhan. And the wet market is embedded in this megalopoly. So it's not a little rural town out in the middle of the boonies somewhere at all. Just to make the point a little further, China is a highly industrialized nation. It's a world power. This is just a few of their Nobel laureates in the sciences, mostly physics and chemistry and so forth, okay? So we're not talking about a little developing country somewhere in other parts of the world. So why did China lock down internally in January, and yet continued to allow people to fly out of Wuhan and other parts of China, even into the summer, even after many countries of the world said, you're not allowed to come into our country. Back then, in fact, it was a big, desperate effort to get foreigners out of China, right? Because even foreigners were disallowed. I remember in particular, Australia became very strict. There were Australian people living in Wuhan were not allowed to go back into Australia, right? And a number of them died from the infection because they were not allowed to go back to repatriate because they were infected. And we didn't know exactly what we were dealing with, but we knew that the death rate was high, okay? So now let's talk about labs, because it turns out that in the same city of Wuhan, just a dozen miles away from the wet market, is a P4 lab. So let's talk about labs. I'm talking about biological containment labs, all right? And there's 4 levels, from 1 to 4. In the European system, they're called BSL, all right? Biological safety lab, we call them P labs, all right, for containment. And so there are 4 levels. Level 1 is the least, and we have our labs up here are level 1. People can walk in and out. Basically, all you need is a lab coat. And if we're going to do some kind of experiment, use gloves. You may not even need a mask or anything like that, or eye protection. Depends on the type of experiment that you're running. All right, it's the minimal lab, P1. Then the biosafety goes up to P4, which is the maximum containment. Nothing is supposed to get in and out of a P4 lab, all right? These P4 labs have positive pressure, meaning that they push air out so that things don't get in, all right? And they wear, the researchers in there wear precisely what is called positive pressure suits. In other words, they look like people from outer space. They look like an astronaut suit, totally enclosed, contained, and they have a tube hanging from the back of the suit that puts air into the tube and some kind of coolant so that they don't suffocate to death inside the suit. You'll see some photographs in a minute, but basically that researcher, that individual is isolated from the lab itself by the suit. Okay, so nothing is supposed to get in and out of a P4 lab. That's the idea, because they have high risk agents, the highest risk agents, Ebola and the coronavirus and HIV and all these highly infectious viruses and bacteria are in these high containment labs. And here is the Wuhan Institute of Virology, known as WIV-CAS. CAS stands for the Chinese Academy of Science. This CAS is equivalent to our NIH, all right? Or the US, the American Academy of Sciences. So this is a P4 lab. It used to be P3. And a few years ago, they put in several $1 million into it and turned it into a P4 lab, just in 2018, okay? And we're talking about an infection that cropped up at the end of 2019. So this is now a P4 lab. This is about a dozen miles away from the Wuhan wet market, on the other side of the Yangtze River. The river that goes through Wuhan precisely is the famous Yangtze, the yellow river of one of the most famous and largest rivers in China. Okay, so what do we have? This lab, this Institute of Virology, boasts, takes pride in being the largest P4 lab, not only in China, in all of Asia, and has the largest collection of viruses and bacteria and of the most virulent type. They have these collections, these libraries of these viruses in here, and they are researching with that. So much so that research leads to publication, right? Not one, but two publications. The first publication came out in 2005 in Science. You know, the two most prestigious journals, scientific journals in the world, Science in the US and Nature in the UK, all right? So in Science, 2005, bats are natural reservoirs of SARS-like coronavirus. In other words, back in 2005, this lab had already identified that the coronavirus was resident in the bats that were being sold in the market. Okay, the horseshoe bat, which is common throughout tropical Asia, and particularly there in China. So they had already discovered the coronavirus strains resident, natural reservoir, it's called natural reservoir, in the Chinese bats, okay? 10 years later, now in Nature, SARS-like cluster of circulating bat coronavirus shows potential for human emergence. You know, potential for human emergence means zoonotic, jumping into the human population, right? 2015. I'll tell you how I became aware of this. We're just two brothers, and we were having a conversation one day. We're both professors. My brother just retired a couple of years ago, political science. And, you know, our father was Italian, so he remembered an article, a news item that was done in RAI, R-A-I, which is the national radio station in Italy, back in 2015, reporting this. And when coronavirus came up now, he remembers, says, he told me, Alfredo, have you ever heard of that report that came out in the Italian news 5 years ago about a coronavirus that had potential to jump from bat into the human population? Because he had heard that back 5 years ago. So I researched it. And sure enough, this is the article. In other words, an Italian scientific reporter, right, picked up this news and reported it in Italy. So now it's international. And the article is there, published in Nature Medicine. Now, let's look at that article a little bit. Here's a review of the article in Nature itself, Nature News, okay, looking like a summary of the 2015 article. 2015, an experiment that created a hybrid version of a bat coronavirus, one related to the virus that causes SARS, has triggered renewed debate over whether engineering lab variants or viruses with possible pandemic potential is worth the risk. 2015 in Nature. In an article published in Nature Medicine, November 9, almost to the date, scientists investigated a virus called SHOC14. SHOC14 is the shock protein, is the one on the spike of the virus, which is the site of attachment with the ACE receptor. The ACE receptor is the one on the alveoli, the human alveoli in the lungs, right? And the shock protein is the one on the spike. That's why it's called corona, because corona means crown in Spanish and Latin. And it's like a crown-like, it looks like one of those Sputnik satellites from the 60s with all the spikes sticking out. Okay, so that's the shock protein, is the site of contact. Horseshoe bats in China. The researchers created chimeric virus. This is called genetic engineering, okay? Made up of a surface protein, the shock protein, and the backbone of the SARS virus. In other words, they took the bat SARS virus that they had identified in the bat population, and they engineered it to put the shock protein on the virus, the sequence, right, genetic engineering, so that the virus, when it went through its infective phase, would generate this shock protein, right? which is the site of infection. So the camera was then tested on human lung tissue, and it infected. So it proved that it could infect human lung tissue, all right? Then they went ahead and tested, so proving that the surface protein, SHOC14, has the necessary structure to bind to a key receptor on the cells. These are the alveoli cells of the lungs, and to infect them. So they're publishing proof that this chimeric virus can infect lung cells of the human lung cells. They also tested on mice live. Why did they test on mice? Because they needed a mammalian model, right? They needed a mammalian model, and sadly, for mammalian model in medical trials, the mouse is the one that gets it because they're being raised in the lab and they're raised for that. You know, before the human gets it, the mouse to test and see at least it can infect the mammal. And then eventually they'll go to human trials with consent and all that. But in mice, it did not kill the mice. So it was a lesser infection, all right? So here it is, it's published. It was engineered. Now, let's look at, so here's the lab, just to give an example of what's a P4 lab. You see the positive pressure suits, right? And these are just sites on the online. I just pulled this from online. That's the whole institute there. Geographically, where it's located. This is the Yangtze River, okay? This is the city of Wuhan, the metropolis, the megalopoly, that goes on both sides of the river. Number of bridges that you can see there. Here's a lab, the Wuhan Institute of Virology, and here's the wet market. And it's about 12 miles from here to here, okay? There's a Google map, if you would like to take the tour. So, one last piece of evidence. How can I copy this? Here we go. So, this is the website of the Wuhan Institute of Virology, which you can read either in Chinese or in English. Sorry? Question, comment? Okay. So here's CAS, Chinese Academy of Science. And here's the Wuhan Institute of Virology. They're very proud of the P4 lab, of course. Okay. You can contact them. Gives a history of how it went from the P3 to the P4, etc. But look at this. This is on their homepage in English. Let's read this paragraph here. I'm just gonna go to the highlight. You can read it on your own if you want later, the whole thing, but I just wanna point out this. The end of the first paragraph. At the same time, it can also effectively improve China's capacity in preventing and responding to biological wars and terrorist attacks and ensure biosafety of China. It's on the webpage. Okay. Responding to. In the meantime, in the international community, we can go to the United Nations for a moment, because for decades now, since the 1970s actually, there has been a commission in the United Nations for disarmament, and it started with nuclear after the Second World War because a number of nations achieved nuclear capacity, right? And then it expanded to chemical weapons and then to biological weapons. So there has been every year a biological weapons convention at the UN, participatory almost every country of the world, including China, on the prohibition, where is the statement? Here we go. Biological Weapons Convention prohibits the development, production, acquisition, transfer, stockpiling, and use of biological and toxic weapons. Okay. Multilateral agreement banning the entire category of weapons of mass destruction. So biological weapons are also considered weapons of mass destruction, which are much more massive than nuclear weapons, because first think of the engineering. To build a nuclear weapon, you need radioactive material, which can be traced. It's documented throughout the world, what countries, what labs have radioactive material. It's monitored, it can be detected at a distance with counters and so forth because of the radioactivity. It has to be highly contained, very dangerous, extremely sophisticated to purify all the centrifuges in Iran and all that. You've seen it on the news, you know, it takes years and years, high technology and all that. And then to transport it, extremely dangerous. You need all kinds of safety. It's bulky, it's large. Even what they call a dirty nuclear bomb would be the size of a large refrigerator, at least, okay? And so forth. And even at the end of the day, that scope would be limited. If it's the size of one of these dirty nuclear bombs, the size of a refrigerator, could wipe out, you know, a stadium full of people or something like that. Thousands, tens of thousands of people, maybe hundreds of thousands. A nuclear weapon, a biological weapon, all right? Biological weapon is a virus embedded in glycerin in a little plastic vial in my pocket. It goes right through security, no detection whatsoever, all right? And if I put that into the water, say into the, I don't know if you've ever been to New York City, Central Park has a huge reservoir in the middle. It's like a lake, you know, just drop that vial in there. So that's the reservoir, by the way, of water for the city of New York. So anyway, nuclear warfare, it's much more gruesome and difficult to contain than nuclear warfare or even chemical warfare, all right? And we have seen the effect because let's say the hypothesis that it was engineered. So then the next question is, did it get out accidentally or on purpose? Or are they actually testing it? Let's assume they're not actually testing. Let's say it was neglect, that the P4 lab is not actually a P4 lab. It's maybe a P2 or P1 where stuff got out, right? And maybe a worker, hypothetically, one of these workers was negligent, grossly negligent about the phase out, how they come out of the lab. First, he was compromised somehow, or had a vial with him or her, who knows, and then went to the market, the wet market, a dozen miles away to eat a bat or something. And he got out into the lab, got out into the market. Not unlikely scenario. But why is it that there was a first inspection of the United Nations to China, and they had extremely limited access to any of the evidence? So much so that the United Nations asked for a second inspection from the WHO, World Health Organization. And the second inspection was a year ago because the whole issue is unsettled. The whole issue of, was it manufactured? Did it get out accidentally? Let's give them credit that it got out accidentally, not on purpose, from the lab, all right? Because they're doing research with this coronavirus in that lab. It's published, The second request of WHO to go back to look at more data, totally denied. Xi Jinping, the president of China, said, no, you were here once before, that's it, you're not allowed anymore, everything is closed, and that's it. So if there's nothing to hide, why not allow people and clear the name of the nation for the world? Why not clear their reputation for the world if it was not manufactured? Come on in, open doors, examine everything, look at all our data, right? So we have to, deduce many things that are plausible with drastic consequences. So I leave it like that. It's my personal opinion that it was manufactured. Now, my only question is, did it get out accidentally or did it actually get out on purpose and they started testing? It wouldn't be the first time that some regime, totalitarian regime, tests stuff on their population. Okay, it's happened before in history. Anyway, I want to shift back to the vaccine because there are a number of bioethical issues there. So
I have a question.
Yes.
Before we move on.
Yes.
So why isn't there like some kind of sanction done on China? Like I remember when the end of World War II, there was a sanction on Germany, with their military. Why isn't there something like that, but maybe like with better regulation on their markets or like certain standards that they should have?
Exactly. All right. It's a very complex question and very complex answer, but let's flesh it out a little bit, all right, just to give some leads. In contrast with the Second World War, where there was abundant evidence, overwhelming evidence, that the Third Reich of the Nazi regime was hell-bent on world domination and the atrocities of the concentration camps, and even internally what was going on and everything. There was, what was it, at least a dozen attempts to kill Hitler by his own generals, right? Because the guy had essentially gone mad. In contrast to all that overwhelming evidence of a rogue regime, here, the evidence is all circumstantial, okay? We don't have the hardcore evidence that the virus actually got out from that lab and contaminated the Wuhan market, which then became ground zero. So it's circumstantial, it's pointing in that direction, but it's indirect. Now you say sanctions. So we would need harder evidence to do a stronger, let's say, international, some kind of complaint, right? In my non-technical terminology, against China. The world community should exactly complain. Now we do have a body, a world body that is supposed to represent all of the countries of the world, and it's the United Nations. It's not perfect, and I take exception to a number of their policies, but they are representing one way or another 200 countries of the world. And precisely the WHO, the World Health Organization that belongs to the UN, went into China the first time several months into the infection, once it was evident that Wuhan was the ground zero and specifically the wet market there, okay? And they were not able to go deep at all, very superficial, and basically confirmed that the infection started there. And that was it. Now, when they asked for the second inspection, nothing, zero, not allowed. But in the meantime, pick up any object that we have today in the US and in the world, and somewhere in the back, somewhere it's gonna say made in China, made in China. This computer that I'm using, the internet, everything is made in China. So China has its products and its services into the world economy. So it's a very difficult, remember when we had the trade wars with China, with the previous administration? President Trump actually started putting some economic screws on China with regards to world trade. But, you know, a lot of the basic stuff that we consume in the world today is made in China. And so it was a very delicate balance how to get China to own up to what was happening there, and at the same time not collapsing the world economy, which in fact to this day we're seeing suffering kind of a tailspin. We're still on the tailspin with regards to the, what do they call that? The chain of production and chain of supply, the supply chain, right? Whole hundreds of vessels with thousands and thousands of these huge containers are still backlogged, held up in ports, not moving through, because indirectly, many people, the great escape from work, right? Many people have decided not to work in public situations. And so the supply chain now is hurting a lot, okay? And so the whole economic issue is very, very complicated, sensitive, and it also eventually is going to spill over into political issues, right? How to deal with the international arena. In the United States, it's a double-edged sword, because in other countries of the world, diplomacy is much more elaborate and more kind of diffuse, and the parliament has some saying in the international policy of international politics of the nation. I'm more familiar with the European nations, but in the United States, it's up to the president. The president is the one who determines international policy and applies it and changes it and so forth. So it's much more unilateral here. There may be advisory, of course, Congress and other branches of government, but at the end of the day, the presidential system in the US is such that the president is the one who handles international politics, okay? So it's very difficult. You got things that are social going into, spilling into, so kind of a sequence here from scientific to social, from social to economic, and from economic to political. And so you see the question is very, very complicated, much more complicated than it was 60 years ago, 70 years ago, after World War II, all right? So my personal opinion is I would like to see something parallel or analogous to the Nuremberg trial that was done back then of the, because since the man himself committed suicide, then his generals and all his underlings, the ones who were able to be arrested were tried there in Nuremberg, and typically they were given life sentences, okay? But time will tell, and history will tell what humanity will do with this. But this, in my opinion, is the first true global pandemic that has affected every nation of the world, and we're still not out of it by a long shot. Kind of a long answer to your question, but it's complicated, right? Okay, sorry, there was a chat here also. Let me see. All right, mice have less receptors. It's not so much the receptors on the mice, but rather the different species, okay? So the receptor is not exactly the correct fit. So it's a little, the fit is not as good, and therefore the infection is not as well. Not only that, number one. Number two, once the RNA is released, the viral RNA is released into the mouse lung, it doesn't have as much virulence as it has for the human tissue, okay? And that's why they tested in mice first, just to see if it would infect a mammalian model. The shock protein that they put on the spike, right? They wanted to see if that would infect a mammalian model. And that's why they used the mice. But of course, the infection was milder. Let's say the infection was flu-like for the mouse. But on the human, that particular engineered virus is much more lethal. So we can also ask the question, what else did they manufacture in that sequence aside from the shock protein? There's speculation that, I remember at least originally there was speculation that the HIV sequence was also injected into was engineered into the coronavirus 19, or maybe the Ebola sequence, right? Because that's your engineering. Remember with CRISPR now, with the technique of CRISPR, we can essentially insert any DNA sequence or RNA sequence of one species into the genome of another species, all right? So the engineering capacity, the genetic engineering capacity that we have today is exorbitant, it's outrageous. And in fact, that technology is fairly simple. We can do it in the P1 lab. Upstairs in our labs, we can do CRISPR, we can do genetic engineering, but it wouldn't be safe. So the level of containment is not the sophistication of the lab itself, it's the sophistication of the containment. But the technology is like cloning, you know, human cloning, to cloning Dolly, to cloning a sheep, or to clone a human is the same basic technique in the lab because it's a mammal cell anyway, right? It's a mammal model. So that's as to that, that post there. The other one, okay, so that goes with regards to the mice. China is a superpower and many ties in shipping that won't allow for studies, in my opinion, exactly. They control many manufacturing and our economy relies heavily on China. Is there a sort of goal post where you don't go past? What sort of benefit would this have for humans that we would need to study, right? So in other words, should we pursue the question? Oh, there's more, something like this, right? In other words, do we let it lie or do we pursue it? Well, everyone was going to have an opinion, you know. Some people may say, well, it's out already. We're kind of getting over the hump globally of the infection. We see, by the way, another pattern that is panning out, which is typical for infections, especially viral infections like influenza and so forth, is what we call a attenuated, a hampered oscillation. Hampered oscillation or attenuated oscillation is that the first wave has a high crest, and the second wave have a smaller crest, and the third wave has a smaller crest, and it attenuates, it hampers out, okay, with time. Similar to the ripples, the wave ripples of a pebble that we throw in a pond. The pond is flat and we throw a pebble in, and at the epicenter where the pebble hits the pond, it will create a large ripple, right? But then that ripple, as it widens out toward the shore, it dampens out, so that eventually when it reaches the shore, the edge of the pond is basically flat again, okay? So it's an attenuated wave, and we'll see that type of pattern, that oscillation, which is typical in nature for epidemics or for infections that come one after the other like that, all right? And so we'll see that happening too. And of course, what's going to continue to help is the vaccines that are developed. And now, by the way, now there are other, well, let me hold on that until we get into vaccines. Let me stick with the question here. I don't know, Chris, everyone has an opinion. Should we let it lie now at this point and not press it further because it could actually be detrimental for the world community? And if we find out, what do we do about it? Well, you know, there's this convention that has been going on in the United Nations for decades now on the prohibition of the manufacturing and stockpiling and use of biological weapons or weapons of mass destruction. So we need to address that, okay? And it is a global, and it should not be only the United States. It should not be US against China, because this has affected the world, 200 nations, right? It should not be the US and China or against China. It should be the whole world community demanding transparency, really. They want to keep their P4 lab, then they have to own up if something happens. So, complicated, right? Okay, you have a follow-up. I'm sorry that we can hear you. Let's see, the control, you did a little follow-up. Here we go. This response ahead was more so for why China was even studying SARS and possibly manufacturing it. I'm not against trying to find answers as to the origins. Okay, I get it, right. Yeah, so we can assume that in the US, our P4 labs are also studying SARS, okay, and Ebola and all these others. But the study is precisely done so as to find out antidotes precisely before the thing gets out, just in case we do get hit with it. So I'm not denying that this kind of research is going on potentially in the US and in several other countries of the world. I wouldn't doubt that several countries in Europe, for example, are doing this kind of research, at least, I would say at least, the UK, France, Germany, Italy, Israel, the NATO alliances, all right, possibly even Australia, maybe India, Canada perhaps. So these large industrialized countries, they have P4 labs and they're doing this kind of research, okay? It's safe to assume that, but the containment is real. The containment, because they have many layers of redundancy. The problem with a totalitarian regime and a P4 lab in a totalitarian regime is that the levels of redundancy can be compromised because of bureaucracy in the policy. For example, another detail that I have found out with Chinese friends and acquaintances, in order to advance in the communist regime of the Chinese government, right, to eventually get to a position of security and prestige and money and so forth in the government, because it's a totalitarian regime. Let's say you start at the level of a governor of a region, like the region of Hubei, where the city of Wuhan is, you know, the governor. So the governor has certain quotas that they have to meet, right, in order to advance in the political structure. For example, they cannot have more than X number of dead people in their hospitals per year. If they have more than an X percentage of the population, let's say Wuhan has 11 million people, they should not have more than X percentage of dead in their hospitals, because the hospitals, every infrastructure is run by the government. The hospitals are government, you know, et cetera, et cetera. So if this mayor, let's say the mayor of Wuhan, has more than 1,000 dead in his hospital per year, you know, then he doesn't get to advance because he's not meeting quotas. Now, these quotas are totally artificial. You know, they're just set up to make a front to say that, oh, city of Wuhan doesn't have more than 1,000 dead in their hospitals, so their hospitals are top, right? But that's not what is actually happening. So what do? They hide the dead, you know, they just don't report it, etc. Misreporting, even in this country, was being misreported. People were dying of cancer in the hospitals, but because they also had COVID, then it was claimed as a COVID victim because the insurance apparently pays more for COVID than they pay for cancer death, all right? And so the quotas were also manipulated here. I'm not saying it wasn't manipulated, but the level of manipulation there, at least we don't have these quotas that are in place in the Chinese regime in order to advance in rank, okay? So you see that the complexity is very, they can lock down the population. They can just tell people, you're not going to go from one city. They prohibit transportation, okay? They have this thing about face imaging. Practically everybody in China is face imaged by cameras all the time. They're doing, what do they call that? Digital recognition, face recognition, right? So everybody's on record by face recognition in the Chinese regime. And so they're not allowed to move from one place to another. They need permission to go from one city to another. That's stuff that is unbelievable to us as a free society, but our societies, we call them open societies, all right? Europe is suffering a lot from that now because their open societies have had blanket borders, open borders, and now it's been just overrun with migrants from other parts of the world that are not necessarily open societies in their own countries. Okay? And they come with their own values and their own ideologies and so forth. and they don't integrate, but they're in, and they're having many children, et cetera, et cetera. So these are the complexities of international politics and the international ethnicities that is going on. Very different system that we're used to. Copyright doesn't exist, right? Anything that comes out, they copy and they make copies. I have many stories, more stories about GRE fraud and trying to get into graduate school and so forth. It's just very, very different from our ethos. The ethos is the ethics of the culture of a time, very, very different from us. Okay? Now, vaccines. Let's round it up here in the next half an hour or so. The bioethics of vaccines. So let's look a little bit at the background. The first three vaccines that occurred, that were manufactured here, Pfizer, Moderna, and Johnson & Johnson. We know that Pfizer and Moderna are the ones that used the novel technology of mRNA, messenger RNA. So they essentially were able to make the vaccine synthetically without having to go through the traditional trial of using the actual virus, attenuated or killed or fragmented, whereas Johnson & Johnson did use the traditional method of obtaining their vaccine. In part, that's why Pfizer and Moderna required 2 shots as opposed to Johnson & Johnson just 1 shot originally, even though now the booster is out. Now, the most troubling part on the bioethics of it was this HEC line, the HEC cell line. All 3 companies used the HEC cell line at some point in their trials, either preliminary trials or the actual production of the vaccine, okay? Without, again, going into much detail, Pfizer and Moderna used the HEC cell line in the preliminary trials just to see if mRNA could work in human tissue, because the HEC line is a human cell line. It's a human cell line, human tissue, right? So Pfizer and Moderna used it in the preliminary stages of testing the mRNA, if it would work or not, before advancing and putting more billions of dollars for research and time and effort into it. And it did work, so they proceeded. Then they did not use the HEC line. They didn't need it anymore because they were using a synthetic mRNA. Whereas Johnson & Johnson, going the traditional route of developing the vaccine, used it also in the production, the actual production of the vaccine. So they're, if you will, a little more tainted, more complicit in this HEC line. So what is the HEC line? Again, another acronym for human embryonic kidney. Well, the word says that this comes from a human embryonic kidney, right? And how do we get that human embryo? How do we get the kidney of a human embryo? Through an abortion. Now, the abortion doesn't generally mean that it was a procured abortion. It could have been a spontaneous abortion. We don't know. The bottom line is we don't know, right? We do know that the HEC line was developed in the 1970s in the Netherlands, but we don't know if it was procured or spontaneous. We know that this HEC line was developed from a little embryonic kidney of a human embryo that was procured by some lab back in the Netherlands in the 70s of human tissue that was going to be discarded as medical waste, because that's normally what happens, sadly, you know, with the remains of a fetus, of an aborted fetus, whether it's a spontaneous abortion that happened in a hospital or a miscarriage, or it was a procured abortion that happened in an abortion clinic. That tissue normally is put in a red biohazard bag and sent to the incinerator, and it's incinerated. That's called biological waste, okay, for health reasons. What happened was that some lab there, back then, obtained this human kidney and probably obtained other organs from this dead human fetus and developed a cell line that became stable, right? So a cell line is no longer the original tissue of the embryo or the adult human, whatever. There's another one that's called HeLa cells that are used in the lab a lot. I remember when I was doing my research and getting my PhD in genetics at Purdue, these cell lines, these human cell lines are used in the lab because it's derived from human tissue and they're stable. But because they're stable, it means that they're actually closer to cancer than they are to normal tissue, right? Because they're immortal. They're immortalized. As long as their passage from one petri dish to another, they keep going for decades, for half a century, 50 years. But it's just an immortalized cell line that is basic human tissue. In other words, it has the human genome, but it's no longer even kidney tissue, right? It's just generalized. And those are cell lines. So you can see that this HEC line is already removed from the original offense that could have been a procured abortion, all right? So the lab that developed, let's go the closest to the abortion possible. The lab that procured the embryonic kidney. That lab, as far as we know, did not participate. We don't even know if it was a procured abortion, but let's assume it was a procured abortion. This lab did not necessarily participate in that abortion. They didn't pay for the abortion. They didn't counsel the woman to have the abortion. They didn't pay for the salary of the abortionist or anything like that. They basically procured the tissue that was going to be sent to medical waste, all right? And the people in wherever it was consented to giving the tissue. Whether they asked the woman who had the abortion for consent or no, we don't know. It was in the 70s. The IRBs were just beginning to come on, and the bioethical committees were just beginning to form at that time, and so forth. So I can tell you from the other cell line that I talked about, HeLa, H-E-L-A cells. Okay, that's another acronym. It stands for Henrietta Lee. Henrietta Lee, and Henrietta Lee was a woman who had cancer, and the cancer was extirpated, and then some lab technician took some of those cancerous cells and derived a cell line from it. Okay, so this technician actually, sorry, not Lee, Lax, Henrietta Lax, sorry. That's Henrietta Lax there, right? And she had cancer, and so they developed a cell line from her tumor, and the big no-no about that is that they didn't ask permission from her. There was no consent to develop that line, but that HeLa line is used all over the world. It's very stable for decades, has been around, but back then it was, when it was procured, it really was, she died in 50s. So this line was developed maybe in the 40s or something like that, okay? And it was, there was no consent from her. She didn't even know what a cell line was or anything like that, but it's a very stable cell line and is used all over the world. So her descendants could make millions of dollars today by just getting 1 cent every time that HeLa cell line is used, right? But it just wasn't a custom at that time yet to ask for informed consent and stuff like that. But that's another one, another example. So a HeLa cell line. Anyway, something similar like this, you know, with this HEC cell line that developed a couple of decades later. So we don't even know if the abortion was procured or spontaneous. Obviously, if it was spontaneous, then there's no ethical issue, right? If it was, unless an ethical issue could have been if they didn't ask consent from the mother of the child, of the embryo, to develop a cell line. Because in today's standards, that should take place, okay? There has to be proxy consent. All right, number 2, the lab, the cell line, once we have the cell line itself, we no longer have tissue from the original embryo. We have tissue that is derived, so it's no longer original tissue, okay? So this misperception that the vaccine is injecting fetal cells into my body from a procured abortion, right, is fantasy. It's not real, it's misinformation. It's all part of that fake news that sadly is going around. What I'm trying to do is place distance, and not necessarily physical distance, because the Netherlands is very far away from us geographically, but ontological distance, a philosophical distance, ethical distance between the use of the vaccine and the original source of the HEC cell line, right? So we have the abortion, we don't even know if it was procured or spontaneous, the development of the cell line that did not participate directly in that abortion if it was procured. Then the pharma that is using this line, that did not even develop the line, they're using the cell line, the HEC line, but they did not develop that line. That HEC line has been around for decades, like I say, since the 70s, right? So this is just one more lab, these pharmaceutical companies, that is using the HEC line, like many, many other labs in the world. In fact, I can tell you, or we can assume safely that because this cell line is so stable for human genome, that many pharmaceutical companies in the world have used this line over the decades. And a number of medications that we're taking probably were tested originally in one of these lines, okay? We don't know, but it's safe to assume that either our HEC or HeLa lines are the two most stable cell lines, human cell lines today in research and trials. So any number of medications that we may be taking may have been tried originally in one of these lines over the decades. So if we go that route, we'd have to start eliminating medications from the counter and from, from the shelves, okay? All right, then what's even further removed is that we're not even a pharmaceutical company that is using the cell line. We're actually consuming the product that is derived from that research. Okay? So we're end users by actually taking the vaccine. So that puts another layer of distance, of ontological distance, of ethical distance between us and the original hypothetically offensive event, which would have been the procured abortion. All right, now, so what kicks in? What is known as cooperation with evil. And this is, we covered this at the end of life lectures, okay, a couple semesters ago. This is one of the principles that we cover, cooperation with evil. We can say, so we have to make this critical distinction between formal or material cooperation, between formal and material cooperation. This is crucial at the ethical level, at the bioethical level, because formal cooperation with evil is just not allowed. We're not the principal agent, but we're cooperating in the sense that we're using a product that came from that, supposedly, let's assume that it was a procured abortion, okay? So the line is tainted ethically. So why is it not formal cooperation? To have formal cooperation, the will has to be engaged. In other words, we need to will the same evil act as the principal agent, to be engaged in formal cooperation with the abortion, desiring that abortion that occurred, hypothetically, 50 years ago there in the Netherlands, okay? Because consider this, we cannot participate in the abortion itself, it's already a completed act. So it's historical. We cannot participate today in a completed act, evil act of yesterday, because it's over. The only way we can participate with that evil act formally is by desiring it. You see? Because the act itself, physically, I cannot pay for the abortion, I cannot take the woman to the clinic, I cannot encourage the doctor to do it, et cetera, et cetera, et cetera. It's an event that has happened already historically. So the only other way that I could participate with an evil act that has occurred historically is by desiring it, all right? But in pro-life, we certainly don't desire that abortion or any procured abortion or any potential abortion for that matter either, you know, even if it's no guilt, we don't desire any abortion, period, as pro-life, all right? And so there's no way that we can engage in that evil act formally, is what I'm trying to say, being pro-life, you know, in a pro-life stance. So at most, we could participate materially. by using the material, in other words, the actual vaccine that derived from the line that came from that embryonic kidney, okay? So there could be material cooperation, but if you remember the lecture, material cooperation is allowed for a serious reason, What's a serious reason? Serious reason is that the virus can kill me. Okay? So I have a responsibility of stewardship. I'll get to that in a minute. But just to remind you of the further breakdown, there is immediate material cooperation and mediate or remote material cooperation, right? And the distinction here is a little technical. It starts getting more technical as we go down the line, but at least at this level, the second level, this intermediate level, this is still important, right? Because for an intrinsic evil act, even immediate material cooperation is not allowed, all right? Immediate material cooperation would be that we are engaged in necessary circumstances. So let's say an abortion. An abortion will have circumstances that are necessary and circumstances that are incidental. What is necessary? Necessary for the abortion are the instruments that are going to be used. So if I'm supplying the instruments, if I'm a supplier of abortion instruments, right, that would be immediate material cooperation. I'm not doing the abortion itself, but I'm a supplier of the instruments that are going to be used, the surgical instruments. Without the surgical instruments, the abortion cannot be done materially. So that's immediate material cooperation, all right? For example, a immediate or remote material cooperation, let's assume that the abortion clinic has a window, okay? And the reason for the window is because natural light come in. Well, FPL, the one who supplies the electricity to the building, and if the electricity goes out, then the abortionist hypothetically could still use the sunlight, natural light that's coming through the window. So the electricity that's been supplied to the abortion clinic is an accidental circumstance. It's not a necessary circumstance, strictly speaking. In other words, the abortion could still go on without the electricity, right? Hypothetically. Therefore, that one is mediate or remote material cooperation, okay? And that one would be allowed for a serious reason to participate. Or the janitor who cleans the clinic is not involved in the abortion in any way, shape, or form, does not desire it at all, but the person doesn't have another job for now. It's the only job that the person has to maintain the livelihood of their family, and so he needs that job, or she needs that job. You know, ideally, that person should be looking for a job elsewhere in a clinic that doesn't do abortions, but may be allowed for material cooperation if there's a serious reason to maintain their family alive. You follow? Okay, so that's material cooperation is allowed. And in fact, we are engaged in material cooperation many times. We don't really know it. When we buy stuff, for example, I use a simple example, is in the supermarket. I go to the supermarket and buy produce for feeding myself, you know, but I don't know if the worker, the farm worker who picked that produce was paid a fair wage. I don't know. But I still buy the produce because I don't want the farm worker to be paid an unjust wage, but I'm not in charge of paying his or her wages. All right, so I may be materially cooperating with that unjust act of the company that is buying the produce at a very low wage, right? Gives a low wage to the farm picker, to the farm worker, and then jacks up the price for the market. But I'm not that company. Okay, so I may be at most a material cooperation, but I don't desire the unfair wage that the worker is being paid. But I do need the produce to stay alive. So there's a serious reason. And we are engaged in material cooperation all the time. In the clothing, we don't know if the brand that makes this clothing is investing in their portfolio in, for example, a a company that makes contraceptives or a company that supplies, does something illegal somewhere, okay? And we still buy the clothing because we don't know where that company is investing their money, right? So we're involved in material cooperation all the time, material cooperation with evil. We don't know it. And yet, but when we do find out, then we have a certain responsibility. If I am buying produce from, I'm just going to throw a name out, Dole or Chiquita Banana, right? And they're paying the workers in Costa Rica pennies for picking the big things of banana, and then they're selling the bananas here at a much higher price. And I find out that there's unjust injustice there. I have a responsibility, an ethical, moral responsibility to write a letter at least to Chiquita Banana and say, pay a fair wage, otherwise I won't buy your bananas anymore, and I'll boycott, and then we can get into boycotting produce, things like that. But it gets, you see how it gets more complicated. In the meantime, I have a need for food, and I need to feed myself and my family, so I still need to keep buying the produce and the clothing. All right. So with regards to the vaccine itself, there's actually a double responsibility, a double ethical responsibility. First, stewardship of self, and then stewardship of others around me. So stewardship of self is that I'm not the owner of my life. I'm not the owner of my life. If I were the owner of my life, suicide would be okay because I decide to end my life. I'm done. I'm getting off this bus. I don't want it anymore. I'm sick and tired of being locked up and not being able to get around. So I end my life. I don't have a right to do that. In fact, it's illegal also. The problem with illegal suicide is how do you penalize the offender, right? Especially if they're successful. And if they're not, they need psychiatric help. They don't need to go into prison. But at any rate, I don't have a right to expose my life beyond reasonable risk, okay? I have a right to protect my life because I'm a steward of my life. And so the vaccine, now there's a vaccine available. It has some level of risk, but it has great benefit. The percentage is very high. And so I have a certain responsibility to take that vaccine. In other words, the burden of proof should be on the people who are not taking the vaccine. And there are legitimate reasons for not taking the vaccine, all right? But it should not be, for example, the hexal line thing, because as I explained to you, it's at most material cooperation and remote material cooperation. My life is worth more than that. My stewardship is worth more than that remote material cooperation. There could be reasons on the clinical side. For example, again, if I'm immunocompromised, or I have a cousin of mine who is actually allergic to some of the substance inside on the vehicle of delivery of the vaccine, you know, it has to be in some substance, some liquid that has all kinds of chemicals in it. And one of my cousins, who's elderly, so she's in the at-risk population, is allergic to some of the substances that are in there, so she can't take the vaccine. And she's been waiting for this Novavax vaccine for a long time, which keeps getting delayed. Apparently the Novavax doesn't have those other chemicals in it, all right? So those are legitimate reasons, but those are clinical medical reasons. They're not on the ethical side of it. And then the second layer of responsibility is responsibility to others, to the people around me, because I don't live in a bubble. I'm related to others. And what happens is with the incubation period being 5 to 7 days, a number of people are asymptomatic. So again, back to the whole issue that they are foci of infection, but they don't know it because they don't have any symptoms. So there's a responsibility, there's a real actual responsibility to others as we relate, even family and friends, to try to minimize that infection and to eventually reach this darn herd immunity, which we can't reach. We're at, in the state of Florida, we're about 50, 60%, which is the same as the county. Another thing too, if you notice, herd immunity works best where? At the most local level, of course, not the global level. The global level, who cares? Because I don't mix with the people of Wisconsin every day, right? I don't even mix with the people of Dade County every day or all of the county or Broward. So it's the most local immediate community. So that's where we need to reach herd immunity at the most local level possible. Dade County, I think we're about 55, 60%. So we don't get to that 70% because of these no-vaxxers, because of the people who have their claiming conscience exception, right, or religious exemption to the vaccine. Sadly, in my opinion, because of all the misinformation that's out there, that I'm getting the cells of a fetus, of an aborted fetus into my body, that Bill Gates has a chip into the vaccine that is going to track the world population, and that eventually it's going to kill a large sector of the world population for population control, on and on and on, all these really fake news that it's out there, right? And you got to know that there's a fear factor that is involved in all this. And so people are using these for excuses. I know that some people are actually getting sick from the vaccine, but that's the same rate as it happens with the flu. Some people who get the flu shot were healthy when they got the flu shot, and they got the flu because of the shot. But it's a very tiny percentage. It's a 0.0001%, you know, something like that. It's the tails of a normalized curve. Okay, it's the tail. The vast majority of the people don't get the actual illness from getting the vaccine. They get the immune response that is necessary for, and it doesn't even prevent, we know that it doesn't prevent getting infected, the vaccine, but it does prevent getting seriously ill on average. Again, I have medical doctors, a number of them you have met through the videos, right? And these people are working today in hospitals here in town, at Baptist, at Mercy, at Jackson, and they tell me, what do they tell me? The ICU units, the intensive care units, are full of unvaccinated people with COVID. So they have COVID, but they're unvaccinated. Now, does the virus discriminate? If we are at 50 or 60% immunity, does the virus discriminate and go and say, oh, no, no, this person is immune, therefore I'm going to skip this person. I'm going to infect this person that is not immune, that is unvaccinated. The virus doesn't discriminate, cannot, doesn't know how to do that. The virus is an equal opportunity infector, all right? It will infect the population as a whole. So what would we expect in the intensive care units of the hospitals in Dade County? 50 or 60% should be vaccinated people who have COVID, you know, if the vaccine doesn't work. But it's not. I'm talking about 90, 95% of the people in ICU units in the hospitals locally and nationally is unvaccinated people with COVID. So what does that tell you? It doesn't mean that the vaccinated don't get it. We get it, we may be infected, but we get mild. We don't go to the hospital, okay? Sometimes so mild that we don't even realize it. All right, so finally, the bottom line about it all. The three things that we have been hearing for 2 years now, all right? By far, which is the most important one? Exactly. The bottom line of the lecture today, existentially, as you leave here, as you go into another building, et cetera, is the washing of hands. The mask, you know, these particles are microscopic. The fomites, which comes out of our mouths, all right, some we can see, some we don't see. You've seen those time-lapse photography of a sneeze, right? It looks ugly. It's a spray, but many of those particles are microscopic. But then again, the load on that microscopic particle of saliva is also tiny. It's a microscopic load. So the viral load of microscopic particles are very small, and the larger particles may get stuck in the mask. But in general, the general idea about the mask is that it helps prevent me, if I'm infected, it helps prevent me from infecting others. In other words, from my infection going out to others, not others' infections coming to me, right? That's the best use of a mask. Different types of mask, how it's worn, everything. it should be the N95 that is more tight around the mouth. If you have a beard, it's not as effective because air gets in through and so forth. So the mask is not that really effective overall. There have been studies. It's more of a psychological thing because when we see a mask, we tend to keep our distance. And the social distance is a little more effective, again, because of the travel of the fomite, which eventually will be affected by gravity. So again, think of fomite is the spray that comes out of our mouth, even when we're talking, not just when we're sneezing or coughing, but even just talking, all right? We're spraying out tiny particles of saliva. When we're just talking, typically they're macroscopic. When we're coughing or sneezing, they're larger particles that actually are visible. But the larger the particle, the more it reacts to gravity and will fall within 6 feet. That's an arbitrary number. It could have been 5 or it could have been 7. But 6 was just convenient. Someone pulled it out of the hat. Let's use 6 because actually it's the distance of 2 arms lengths between people and people. Okay, so we have about 3 feet more or less from our face to the tip of our fingers, the average person, and 3 more on the other side. We'll put 6 feet between mouth and nose of 2 people. So that's kind of a convenient 6-foot measurement, all right? But it could have been 5, it could have been 7, who knows, who cares? I also want to point out that calling it physical distancing is really a misnomer. And again, this is a conversation with my brother as a social scientist, is in tune to these things. If anything, it's not physical, it's not social distancing that we're looking for, it's actual physical, material physical distancing, all right, of X number of feet. But social distancing is very different. Social distancing is closing in on oneself. If anything, we need social proximity. Virtually, of course, but social proximity. I'm talking about the height of the pandemic last year. This physical distancing also locked us up and locked us into ourselves. And we became drawn in, and that was detrimental, particularly to the youth, because in the formative years, that's when they're learning how to socialize. And that year was very detrimental, not being able to go to school, locked in with the canvas thing. And so naming it social distancing was a misnomer from the get-go and has had sociological implications to the negative, all right? So I try to, whenever I remember, I try to use physical distancing instead of social distancing. And if anything, we should make an effort to be socially closer to each other by staying in touch, by phone calls, by conversation, by text and virtual and all that stuff, but staying more in touch with each other than before. But the washing our hands, at least 20 seconds. Why the 20 seconds? With normal soap. Don't use antibacterial soap because that's just making more mutants of superbacteria. The bacterial soap is what percentage effective? 99.99, right? The antibacterial, 99.99. What does that mean? That means that the 0.001% of bacteria become immune to that soap because they're mutants. But now that you wiped out the other 99.99% of the population of that bacteria, you have given ample terrain, you have opened up a niche for this 0.001% bacteria that are immune to that soap. You have just cleared the ground for them to grow galore because they have no competition, because it just got wiped out by the antibacterial soap. So actually, unwittingly, with this bacterial soap, or wittingly in the industry, we are just encouraging the superbacteria, the more immune to more stuff, to continue to grow and flourish.
What about the hand sanitizer?
Sorry?
What about the hand sanitizer?
Hand sanitizer strictly for the virus, yes. All right, but it's specific for the virus, to contain the virus in lieu of the herd immunity thing. So for now, yes. But the antibacterial, because it's more broad spectrum. Okay. Plus it's bacterial anyway. There's no antiviral. It would be so harsh that it would actually damage our skin. Okay, but the 20 seconds, why at least 20 seconds? 20 seconds. And normally, I can tell you folks, we normally don't wash our hands for 20 seconds. We do 5 to 10 seconds and then we're out. And we're not thorough as if we were going to go into surgery or something like that. because it happens that the coronavirus, in addition to the protein coat, the protein shell that it has, right, the structure, it also has a membrane, a bilipid, a lipid bilayer, which is a standard lipid bilayer of our cells, all right, the phospholipid bilayer of any cell membrane. And that is on the outer layer, on top of the envelope, or the protein envelope. And what happens with that lipid, what's a lipid? It's just a scientific name for oil, fat, grease, wax, all right? Gets dissolved by soap, because soap is amphipathic, right? It's got polar on one side, non-polar on the other. These guys are non-polar, so they get dissolved. The lipids get dissolved by soap. That's why we have greasy hands, oily hands, we wash with soap, and they come clean. So that's the whole thing. It's very simple. The soap actually begins to dissolve the lipid layer, which prevents or reduces the possibility of the virus to infect, to lodge on the alveoli, all right? So it hampers, when we get rid of the lipid layer on the virus, it hampers its ability to infect the alveoli. In other words, that lipid layer is part of the attachment mechanism. And so when we dissolve that, we're actually preventing the virus from infecting. But 20 seconds at least, to allow the soap to work, because we're going macroscopically, super macroscopic, right? Keep in mind, look at our skin, you know, we see tissue, we don't see individual cells. So individual cells are macroscopic. Thousands of bacteria fit inside a single cell, and thousands of viruses fit inside a single bacterium. So we're talking about super macroscopic structures here. And that's why it takes some time physically for that soap to work its way into the virus and start dissolving that lipid layer, right? So that's the reason for that. And so that we can reach this at some point. And now, you know that there's some other products that are coming out in the market, beginning to come out, pills that are having some effect because now the research is more robust and is moving forward. So they're little by little, there are alternatives coming out to the vaccine as preventive medicine. Doesn't say that if we have the virus, it's gonna kill it or anything like that, but at least preventive, which will help us somehow reach this herd immunity. So by hook or crook, we need to get there at some point in the human population to walk away from the pandemic. But it's still not over yet. We seem to be over the cusp, if you will, and beginning to go on a negative slope. But depends country by country. And as long as this thing is around in some country of the world, it still has the potential for generating a new variant, and from the variant eventually a new strain, and starting the loop again, right? I just heard this morning in the news, Austria, a very highly developed country of the world, Austria is locking down, the whole nation is locking down because they have a new wave now, new surge. So it's not over, like who was it? The Maggio, it's not over till it's over? Or some famous pelotero, baseball. It's not over till it's over, and therefore we cannot lessen our guard. Please wash your hands as often as possible for 20 seconds, at least 20 seconds. Folks, that's all I have to say. Said a lot today, but you can see that at least it's relevant. We're using the COVID as a case study of pandemic bioethics. And that's it. Questions, comments? No summary for this one. Aren't you happy that you've been doing dozens of summaries already? So at this point, I don't want to see another summary. It was long, yes. Yeah. So, well, you're done with the summaries now. For this and for the last one, no summaries. Okay, another chat here. Let me move it up. Okay, my pleasure, Christopher. Thank you. Thank you all for participating. God willing, we'll do this one last time. And December 4, I want to make an emphasis to please do try to come in person, because I'm also getting evaluated that time for the whole program by the chair of the department here. And so it's going to be on transplant. I always like to finish with a happy topic, and it will be on transplant organs, transplant bioethics. So stay tuned for that. All right. Okay, let me close this. Don't forget to finish up your hours. A couple of you still have a summary pending, and a couple of you still have some hours pending. I'm still offering the 3-for-1 if you need to close that up. Okay. So stop recording.
Thank you, Professor.
Okay, my pleasure. Thank you all. We'll see you in a couple of weeks.
Bye-bye.
Oh, and a happy and blessed Thanksgiving to you and to your families. I hope you have a little time off there to regroup and to enjoy the break, okay? Thank you.